Welcome to the DSM Biotechnology Center in Delft, the Netherlands. The DSM Biotechnology Center is the starting point for the development of fermentation-based processes for the various market segments. It employs approximately 500 people, equally distributed over 5 departments: genetics, fermentation, downstream processing, applied biochemistry, and analysis. This short introduction will highlight the capabilities in each of the technical disciplines and will provide insight in the way the center interacts with the various production sites all over the world. New processes are developed in teams with representatives from each of the technical disciplines. From the start of the project, the team includes specialists from both the Biotechnology Center and the production facility who are experienced in transferring processes to large-scale production. This organization ensures that there will be an optimal fit with the existing infrastructure at the production site. The genetics department covers all aspects of microbial strain construction, selection, and improvement. Technologies applied range from classical strain improvement to directed molecular engineering and synthetic biology. DSM has extensive experience with a large variety of microbial strains and product categories. The plug-bug concept offers a versatile platform for protein expression, using established, high-performing strains that can almost instantaneously be introduced in large-scale production. As for genetics, the fermentation capabilities cover a wide array of microorganisms. Fermentation processes have been developed for multiple strain product combinations up to and including large-scale commercial manufacturing. Fermentation scales at the DSM Biotechnology Center range from half a liter to 3,000 liters. However, most of the fermentation process development is performed at 10 to 15 liter scale. More than 60 identical setups ensure that all fermentation scientists speak the same language in terms of process parameter analysis. In most cases, scale-up to production is realized in one step, that is, from 10 liters directly to 25 to 100,000 liters. Downstream process development capabilities include all common unit operations for solid-liquid separation and the various purification approaches like chromatography. All unit operations at the DSM Biotechnology Center are downscaled versions of the production site infrastructure, thus enabling efficient performance evaluation and process transfer to the production organization. It is in the area of fermentation and downstream processing that transfer to production is addressed by a team of experienced technologists, with intimate knowledge of various production sites and with frequent personal contacts with key personnel of the production organization. The analytical department is an all-important part of the DSM Biotechnology Center. It plays essential roles in strain selection, fermentation, and downstream process development. For production sites, analytical methods are developed, transferred, and implemented. As applies to the other departments, the analytical group has an important additional function in supporting running production in, for example, troubleshooting exercises. Next to the more standard analytical tools, the infrastructure encompasses a range of advanced instruments. Various mass spectrometers, including a Synapse G2S, and NMR facilities up to a 700 MHz instrument. Several mass spec and NMR instruments are equipped to serve high-throughput experimentation in the various R&D departments. Mass spec and NMR are used for the identification and quantification of different classes of compounds, like proteins, small molecules, carbohydrates, and lipids. DSM has extensive experience in detailed molecular and biochemical characterization of proteins. Analysis include identity and purity of the target compound, impurity analysis, intact mass amino acid sequence determination, and the range of protein modification. In troubleshooting mode, the approach often involves a first diagnosis by NMR, from which an analytical strategy is developed to monitor the solution. A responsible scientist represents the analytical discipline in project teams and coordinates all analytical efforts required throughout the project lifecycle. An expert forum in weekly meetings discusses analytical project questions or production problems and suggests analytical solutions. The efficacy of DSM's large-scale production capabilities is to a large extent based on the long-term analytical experience. The extensive infrastructure exemplifies that analysis is considered a cornerstone of DSM's biotechnology ambitions.